It's time to see the naming, origin and nomenclature of aminoglycosides. There are two genera of actinomycetes which are the source of the aminoglycosides, and first of all they have three suffixes: mycin, mycin and sin. Sin is very easy, amikacin. It's a semi-synthetic aminoglycoside and it should not remind us of Johnny Sins. Okay, this amikacin. Okay, now coming to mycin: gentamicin, nefilmicin. What drug is this? Cecomycin, plazomicin. And these are mycin: streptomycin, tobramycin, kanamycin, neomycin, paromomycin. These are obtained, all these mycin are obtained from Streptomyces genus, and this mycin, it is obtained from Micromonospora. Okay, and these are the genera of actinomyces which reside in soil bacteria. तर इथं काय सांगितलं आपल्याला? मायसिन वाले स्ट्रेप्टोमायसिस बॅक्टेरिया मधून मिळालेत आणि मिसिन वाले मायक्रोमोनोस्पोरा बॅक्टेरिया जीनस पासून मिळालेत आपल्याला. आणि अमिकासिन सेमीसिंथेटिक आहे विथ सिन सफिक्स आणि त्याचा सोर्स ग्राम पॉझिटिव्ह सॉइल बॅक्टेरिया actinomycetes. नंतर काय लिहिलं हे सगळेच लिहिलं स्ट्रेप्टोमायसिन, कॅनामायसिन, निओमायसिन, पॅरोमायसिन. मायक्रोमोनोस्पोरामधून आपल्याला काय काय मिळतं? जेंटामायसिन, सिसोमायसिन, प्लाझोमायसिन. मिसिन ना सेमीसिंथेटिक अमिकासिन मॉडिफाईंग कॅनामायसिन, म्हणजे कॅनामायसिनला मॉडिफाय करून मिळाले ते. आणि फर्स्ट डिस्कवर्ड अमिनोग्लायकोसाइड हे स्ट्रेप्टोमायसिन होतं, जे की स्ट्रेप्टोमायसिसपासून मिळालं होतं. आणि फर्स्ट अमिनोग्लायकोसाइड डिस्कवर्ड फ्रॉम मायक्रोमोनोस्पोरा इज जेंटामिसिन. Now it's time to see the nomenclature difference between aminoglycosides and macrolides. We know the mechanism of action of aminoglycosides and macrolides, where they work. Next thing to be seen is regarding the how to differentiate and what are the sources and what are the properties of macrolides. All this is to be seen. Now talking about the aminoglycosides and macrolides, they have the same suffix called mycin. But how do we differentiate them? This is one fact to be known, that patients with cystic fibrosis, they inhale tobramycin, which is an aminoglycoside. Kanamycin and neomycin, they are other aminoglycosides. Okay. Now the macrolides, they are erythromycin, which was the first macrolide that was discovered, and others are clarithromycin, azithromycin, rosithromycin. So here the suffix is thromycin. म्हणजे माझे सीन फेकून द्या, thromycin. Okay, and these are all macrolides: erythro, claritro, azithro and roxin. Eri, clari, azi, roxin. All these drugs, the macrolides as well as the aminoglycosides, are derived from actinomycetes, but aminoglycosides are derived from Streptomyces genus, whereas the macrolides, they are derived from another genus, that is Saccharopolyspora erythrae. So, where were we? Azithromycin, it's most commonly used drug in the world. Gentamicin, netilmicin, cecomycin, plazomicin. Okay, these are all what? These are mycins, and they are the aminoglycosides. And we know from where they are derived from. So, erythromycin, parent drug, clarithromycin, and then came azithromycin. They are modified forms of erythromycin. They all inhibit the protein synthesis by acting on the 50S ribosome, and these are all macrolides. Even though they have the same mechanism of action, but they have different pharmacokinetic side effect profiles and drug interaction. Oral erythromycin, it's not enteric coated. It gets destroyed by the gastric acid and produce GI irritation. Bioavailability half-life of erythromycin is less, as it gets destroyed by the gastric acid, and more frequent dosing is required, which reduces its compliance. And clarithromycin, azithromycin, they are acid-stable drugs. Their enteric coating is not required in them, and absorption, bioavailability, half-life, tissue penetration, they are better for these drugs. And these drugs, they penetrate the bacterial cells better. What these azithromycin, and azithromycin even enters the macrophages at high concentration. Less frequent dosing is required for clarithromycin, and azithromycin is given OD only. It's available in extended formulation. Erythromycin, it mainly acts on aerobic, okay, gram-positive bacteria. Clarithromycin and azithro have a broader spectrum of activity. Azithro, clarithromycin कमी few side effects as compared to erythromycin. Erythromycin's side effect is on the heart rhythm, long QT elongation and arrhythmia. And its cardiac side effects, they are reduced with azithro and clarithromycin. Erythro and clarithromycin are notorious for causing drug-drug interaction. They affect the function of cytochrome P450, which is used. ते जे cytochrome P450 असतं ना लिव्हरमध्ये, तर त्याचं फंक्शन ते अफेक्ट करतात. आणि these are used in many other drugs. These drugs should not be prescribed in liver pathologies because they can be hepatotoxic and interfere with cytochrome P450 function to produce drug interaction. Azithromycin, त्याचे minimal interaction with cytochrome P450 आहे. Aminoglycosides, macrolides, they ribosomal protein synthesis machinery वरती काम करतात. We know that, and they reduce the protein synthesis in susceptible bacteria. जे aminoglycosides and tetracyclines आहेत ना, they act on the 30S ribosome unit. सो ह्याचं आणि जे क्लोरामफेनिकॉल, इरिथ्रोमायसिन, लिंकोसामाइड आणि लिनेझोलिड, ते आहेत ना, ते 50S रायबोसोम युनिटवरती काम करतात. आणि याचं एक सरांनी आपल्याला हे सांगितलं होतं, ट्रिक सांगितली होती की बाय ॲट 30 आणि सेल ॲट 50. बाय ॲट 80 मीन्स अमिनोग्लायकोसाइड्स अँड टेट्रासायक्लिन, वेरस सेल ॲट 50 म्हणजे सेल CELL म्हणजे क्लोरामफेनिकॉल, इरिथ्रोमायसिन, लिंकोसामाइड आणि लिनेझोलिड. आणि ही गोष्ट लक्षात ठेवणं गरजेचं आहे की जे अमिनोग्लायकोसाइड्स आहेत तर ते आर बॅक्टेरिसाइडल, वेरॅज मॅक्रोलिड्स जे आहेत तर ते आर एरिथ्रोमायसिन, आणि जे मॅक्रोलिड्स आहेत तर ते आर बॅक्टेरियोस्टॅटिक. सो हे वाचून घे सगळं. दिस इज व्हेरी क्वाईट इझी चॅप्टर आहे. ओके, so buy at 30 and sell at 50.